eminent participants, and my amazing colleagues and friends of science, research, and innovation. A warm good morning to everyone, and I'm highly delighted to welcome you all on behalf of Tata Institute for Genetics and Society for this webinar on challenges and advances in SARS-CoV-2 research. This webinar is a part of TIGS Science Serving Society program. Before we proceed, I would like to introduce myself. I am Arunachalam Ramaiah, Senior Scientist in the Tata Institute for Genetics and Society. I'm glad to organize and be the moderator for this webinar. Very excited to have a total of 650 participants from India and overseas registered for this event. We all know the current COVID-19 pandemic has underscored the pressing need for countries to focus more on elevating scientific research and innovation. And in this context, this webinar will provide insight and shed light on A, past ongoing COVID-19 pandemic and future pandemics, B, host molecular and immunological responses to coronavirus, C, clinical intervention based on genomic surveillance of SARS-CoV-2 virus and COVID-19 big data, challenges in India had faced during the early 2020 and how India overcome this issue, how the recent developments in India are critical in diagnostics, prevention, and treatment of COVID-19. Distinguished ladies and gentlemen, today we have 6 prominent scientists from both India and the United States to share their research experiences. 3 eminent scientists, Dr. Shashank Tirupati from Indian Institute of Science, Bangalore, Dr. Vinod Skoria from Institute of Genomics and Integrative Biology, New Delhi, and Dr. Vaithilingaraja Arumugaswami from the University of California, Los Angeles, are here to deliver the invited talk. 3 renowned scientists from Bangalore are here as panelists. Dr. Chitra Pattabhiraman from National Institute of Mental Health and Neurosciences, Dr. Varsha Sridhar from Molecular Solution Care Health, and Dr. Uma Ramakrishnan from National Center for Biological Sciences. I strongly believe this webinar will help us accelerate in exchange ideas and scale up our research activities. I'm also confident that you will find new ideas, fresh energy, and novel collaborator to sustain your efforts to recover the world from COVID-19 through science and research. I take this immense pleasure to welcome and hand over the session to fantastic colleague and director of Tata Institute for Genetics and Society, Dr. Rakesh Mishra, to chair the session. He is a J.C. Bose National Fellow and an elected Fellow of the Indian National Academy of Science, Indian Academy of Sciences, and National Academy of Sciences. We are honored to have you as a chair for this session. I wish you all a very successful webinar. Over to Dr. Rajesh, please.
Thank you. Thank you, Arun, for a very nice introduction, and thank you for taking initiative in organizing this webinar, which is one of the privileges that we have generated during this pandemic. I'll be very brief, and then I will like to listen to fantastic speakers and panelists in the program. Just to add a few things, the theme of today's webinar, the challenges and advances. I think all of us agree that in past 1.5 years, we have seen some amazing, unprecedented advances against the challenges this humanity has faced in centuries. And science has played a major role in various aspects of this disease. The first thing that came beginning of last year was what is this disease and how to find out the diagnostics. And that was such a big challenge. And this diagnostic is very sophisticated. It's not a simple thing. You have to take RNA. Those who do molecular biology know how tricky it is to handle RNA, and then convert into DNA, then amplify specifically and tell supply of these expensive chemicals and the infrastructure to do this kind of sophisticated tests at a very large scale was such a challenge. Country like India, it was just impossible to imagine to reach where we are today. We have almost overcome all these problems. We are totally endogenous. The cost of test has gone 20-fold less compared to what it was. Hundreds of labs, the government organization, private organization, hospitals are doing the tests left, right, and center, and even common public. Everybody knows what is CT value. Many scientists will have difficulty in figuring out what is CT value. So they are not happy with plus or negative or positive. They want CT value of their test. So that's something amazing that we have achieved in past year or so. And to add that, even the new tests have been developed and implemented very, very successfully, which have actually changed the scene. Vaccines. I was the one who used to say that this pandemic will be fought without vaccine. And I'm so happy to be wrong. And I was saying, based on traditional approaches, that no vaccine comes in less than 5 years, 10 years time. But today, here we are. We don't have one, two, we have many vaccines. And one of the most revolutionary thing, I think, which will go well beyond this pandemic, is the mRNA vaccine and therapeutics, which have come out. And in a way, thanks to pandemic, that even regulatory bodies have become more accommodative and realizing the need, and that has really proven so successful. So this is the challenge which scientific community, industry, and regulatory bodies have really faced very well. In the area of drug and taking care of the disease, while drugs are not there yet in a real sense, but we have learned a lot how to take care, and mortality has decreased significantly compared to what it was in the beginning of this pandemic. One challenge which we are still facing, and that has lots of other input, that's why it's a little bit difficult, is the communication and conveying people the correct information and taking correct steps. Because we know this pandemic can be controlled not only by vaccines or drugs, people have to participate. Then only we'll be able to fight it out. And that is what something we need to continue. And this kind of seminars and effort, I think, are a important contribution to this effort. Surveillance of the disease, to what extent the disease is, whether it's spreading or not, whether the new variants are coming or not. You're going to listen many of these things in more detail. But it's just amazing that how sophisticated technology at large scale data have been so quickly assembled, and we are able to follow the disease from sequencing data that is being generated. And not only that, even from the wastewater, we can now figure out the extent of infection and even the emergence of or the presence of new variant, or the situation of how many variants and what kinds of there, just from the sewage water. So that is something amazing, and I think is going to go a long way in public health system in handling infectious diseases. And just to end, how we prepare ourselves for the such pandemic or such disaster in future. I mean, there are lots of discussion going on. The key thing we have probably not emphasized enough is that from where this disease has originated and why this has happened can be prevent this. And that is something I think we have to evolve. We have to learn that if we interfere or encroach the nature, which part of the nature which doesn't belong to us, we are going to be in the trouble. If we encroach the wildlife, we are going to not only trouble them, also invite new diseases like this, because we are crossing our limit. We have to learn that this planet belongs to not only humans, it belongs to all the living beings on this planet, and they are all equally important. This is something which we have to learn for long-term survival. And I think this message should come out when we are out of this pandemic, hopefully in coming few months. So I'll stop at that, and I'll look forward to listening to our fantastic speakers and panelists. So over to you, Arun.
Yeah, thank you very much, Dr. Rakesh, for your warm opening remarks. Now we are moving to the invited talk session. I request the participant to post your question in the QA section for the speakers. With the great pride and honor, I'm here to introduce the first speaker of this session, the man of the hour, a popular virologist and stem cell biologist from the United States of America, whose research focuses on understanding pathogenic mechanisms of diseases caused by pandemic potential RNA viruses, including corona, dengue, Zika, and chikungunya viruses. His laboratory is actively working on developing pan-antiviral agents and vaccine strategies, and has established several cells, organoid, and animal model systems. He has received his Master of Veterinary Science from Indian Veterinary Research Institute and PhD at University of Arkansas, United States, followed by postdoctoral fellowship at University of California, Los Angeles. That's Dr. Vaithilingaraja Arumugaswami, Associate Professor from the Department of Molecular and Medical Pharmacology at University of California, Los Angeles. Dr. Vaiti will talk about COVID-19 and future pandemics, research approaches, and challenges. I welcome you, Dr. Vaiti.
I would like to thank Arun first, because he's my colleague and a very good friend. We have a long history of collaboration in the past 8 to 10 years. We started off with Ebola and influenza in 2013 or 14. At that time, our work was very small, but we were making our foundation. Then we evolved into studying Zika virus when 2016 outbreak happened. So we published paper together. Arun was the first author in that paper. Then subsequently the pandemic hit. So I'm going to share some of the early days of our work, and also what we learned, where we can go from here. And also I thank Dr. Mishra for spearheading this webinar. As he mentioned, the positive side of this pandemic is we started using this Zoom call or any webinar-based platform. The only thing, when I started using the platform in the beginning, humans are not attuned to watching their own face when we are speaking. So it is very interesting. We start adjusting this new normality. And also I thank Dr. Dasgupta for communicating all the details during this past few weeks. So with that, I'm an associate professor at Department of Molecular Medical Pharmacology, David Geffen School of Medicine. So here in this talk, we present some of our early data and also our ongoing research. And all these opinions are not reflective of UCLA or any entities. And also I do not have any conflict of interest in this presentation. So just to get to this, our current story, we in early January last year, my lab, essentially pre-pandemic, we were focusing on Zika virus, studying Zika virus. Then in January, we kind of everybody heard, at least some people heard, there are 400 cases of SARS-like symptoms in Wuhan, China. 8 people died. So for me as a virologist, it's a red flag. For rest of the world, it's not. It's happening somewhere in a corner of the world. When we were studying Zika virus, there was not much mortality in the adult patients. Of course, the young or developing fetus were heavily affected. So to me, this was a red flag. And immediately I talked to Arun, here your own Arun from TGIC. At that time, he was a scientist at UC Irvine. So we spoke very quickly, and we said, let's study this virus a little bit more. Let's do a detailed genomic analysis. So Arun did all the heavy lifting within, it's all started in January, I think January 22nd. Within a few days, he pulled together all the available genome. At that time, in the beginning, we got only one genome. Then subsequently, January 25th, we got 45 genomes. So Arun quickly analyzed and narrowed down the current SARS-CoV, or at that time, it was called 2009 novel coronavirus. So he narrowed down the origin of this virus. It's related to a bat coronavirus. So we submitted this paper while we were waiting for the review. We submitted this paper to bioRxiv. As Dr. Mishra was saying, there are a lot of misinformation at that time. So, but this paper received huge attention, thousands of views. And we essentially nailed the origin of this virus. However, we still don't know how this jumped into the human. So that's a major issue. Another thing, as a virologist, you would always, not only virologist, anybody studying infectious disease agent, they know the host pathogen balance, the interaction is crucial. If a virus or pathogen can quickly kill the host, it's not a good pathogen. So that you could see from SARS-CoV and MERS-CoV. It's a high fatality rate or mortality after infection. However, this current SARS-CoV-1 or CoV-2, it's very smooth. It's very stealthy. A lot of time people don't even take care because they think it's a mild flu, right? However, the susceptible people, right now we see several million people died, and the pandemic is still ongoing. Of course, we have vaccine, but the vaccine distribution inequality, and also people refuse to get vaccinated. So this virus is going to be here for a while. So now many of the countries adjusted to live with the COVID. My daughters, they were online class for last 1.5 years, but last month they started the school. So now in Los Angeles, at least, we have more rhinovirus outbreaks. So it's a scary thing for parents. You know, the kids are sneezing and coughing, have fever, but it is not COVID, it's a rhinovirus. So that's where we stand. Here, Dr. Tripathi would explain more about the genomic side. However, one thing you could see, any of these virulent viruses or stealthy viruses, they pack additional genes in the genome. You would similarly see with the HIV. They carry extra genes or in their genome to control the human cellular machinery. So when this virus was essentially contained in Wuhan in 2020, of course the people flying out through Wuhan, and also the virus already started spreading throughout the world. So we got the viral strain in March 13, 2020 from CDC. At that time, many people don't want to, they are scared to work with this virus. My lab was almost first in the East Coast, United States, to culture this virus. We started immediately doing the drug testing. On the right side, so you see me with a space suit, and I still go there time to time. Now we are culturing all the variants of concerns and testing them with multiple drugs. So here, my lab actually did a lot of work on the vaccine and also virology side. But when this virus, the pandemic started, the UCLA collaborator, Dr. Robert Damasiak, we kind of hit it off, and he's running a drug screening facility. We immediately started screening drugs. So within a week, we started identifying drug compounds. So this group, we have many of the non-virology people. So I would like to give you this cartoon. It was originally made by undergraduate students in my lab, but we kind of polished it. Here is a mammalian cell and a virus. The virus can enter through the cells through a cell receptor. In case of SARS-CoV-2, it's ACE2, human ACE2. When the virus injecting the genome into the cytoplasm, the genome gets translated, made multiple proteins. Then the genome actually replicating in a specialized compartment inside the cells. Once enough genome and also a lot of proteins being made, they got assembled and you get a new viral progeny is coming out. So a single cell can be infected, once infected with a single virus can have thousands of viral progenies coming out. So that the new virus can be coughed out or different modes can spread to a susceptible host. So we could actually stop the virus at the entry level. A lot of monoclonal antibodies work at this blocking the entry. And also when people are getting vaccinated, the antibody generated from the vaccine response can also block the virus from getting inside. So that's how you get the protection. And also you can stop the viral translation or genome replication and also the virus coming out. So multiple points you can attack the virus. If you are attacking at the step 4, the cells are already sick. So it's better to attack the virus at the early stage. So we at that time, we kind of tested whatever the immediately available drugs. We tested interferon beta and also hydroxychloroquine. The hydroxychloroquine can work to some extent. However, the virus can use different mode of entry, so this drug cannot work. So there was a lot of controversy about using this in the frontline worker. But the current variants, the Delta variant, the hydroxychloroquine doesn't work. Okay. So we have this system established, and also in the beginning, we all thought the SARS-CoV-2 is only infecting the lung and causing the lung disease. However, we started learning the virus can affect multiple organ systems. So the first thing for a virologist to do is to grow the virus in a quantities we can test antiviral compounds. So we were testing multiple cell types. All the red ones were not getting infected. Only the green cells were infected. So these are actually cancer cells or are kind of not a good cell types to study the mechanism of virus replication. So we actually fortunate to work with Brigitte Gomberts. She's a pediatrician, MD, in UCLA. She has a lung air liquid interface model where the lung stem cells from the trachea or the bronchi are collected and grown in a cell culture. So we use this model to first study the SARS-CoV-2. So the green is the SARS-CoV-2, where the red or the acetyltubulin, these are the fine fimbriae or the cilia responsible for pushing the mucus out. So this is a very early study. At that time, they didn't have this kind of data. The virus can actually infect the ciliated cells. So you could see that. And also here, you could see the cilia movement. It's kind of a beating cilia. However, with infection, you could barely see any of the cilia beating. There's one cell around here you could see beating, but without infection, there's a healthy cilia beating. And also, we worked out the distal airway organoid system. This paper published a few months back. And also, currently, we are using microfluidic system. It's a microphysiological system where you can load the cells in a microtubules channels and study SARS-CoV-2 replication there. And also another thing, early on, we at that time, people were saying, okay, people have patients with underlying heart disease or hypertension, they are prone for more severe COVID disease. So there's no other information available. So in collaboration with Clive Swanson at Cedars-Sinai, we were the first group to show that SARS-CoV-2 can infect iPSC-derived cardiomyocytes. So here, these are all the human iPSC-derived cardiomyocytes. Here, you could see it's stained with the cardiotropinin. It's a specific marker for cardiomyocytes. With infection, you could see a lot of viral replication. And this paper was published, and this is one of the highly cited paper in cardiology field, hundreds of citations. And also we were highlighted with in NIH site for this contribution. And also you could use this model to study the SARS replication. So on the top, you could see the heart is beating. This is cardiac cells in a dish. So without infection, the cells are healthy and it's beating. But here, there's no beating, so the heart is dead. So you could model the SARS-CoV-2 pathophysiology in this model system. And once we have all these systems in place, we studied a library of chemical library of kinase inhibitors. A human body, our genome encodes about 450 kinases. So 430 of them are druggable. So we took this kinase library. Here is our collaborator, Robert lab. He's running a big screening facility, and he's the guru for us for the drug development. So in the first primary screen we did in Vero E6 cells, we identified 34 kinases. Then we subsequently tested them in verified one compound. It's a berzosertib. This compound is actually owned by Merck, Germany. So here, wherever you see red, if you see a lot of red, means more replication. This from our initial screen. Berzosertib with various concentration, less red, means it is inhibiting the virus. Same way, there is a mTOR pathway compound also can inhibit, but however, all the mTOR compounds we identified, they're also very toxic to the cell. So the only way we could get the compound to the clinic is it should be non-toxic. The berzosertib is already in the clinical trial, phase 2 clinical trial. It is safe in the human. It's essentially a cancer drug. So we are currently working on a pre-IND package to get this drug to the patient. Here it's just a kind of a network analysis. So here essentially the iPSC cardiac model, where both the wells are infected with SARS-CoV-2, but this well received the drug berzosertib, so it still maintained the heart beating. So here, without drug, the cardiac cells are completely wiped out, and the cells were all dead. Here it's a little more molecular analysis of the mechanism or mode of drug activity. This is a ATR kinase inhibitor, so we do see some inhibition with adding the drug. You would see the viral the phospho-CHIK inhibition. So here it's this slide was put together by Dr. Arun. It's a lot of effort just to show SARS-CoV-2 infection is regulated by multiple signaling pathways. Here it's a ATR, ATM pathway, and also mTOR pathway. So we got a lot of funding, millions of dollars of funding for this research effort, and we got another big grant for $4 million grant for studying the metabolic side of the SARS-CoV-2 disease progression. So and also we set up established a mouse model. This is based on human ACE2 expressed by adeno-associated viral vector. So this is a systemic model. This study also published in last year in JCI Inside. So essentially, the red the animals, we had like 8 to 10 animals. The red animals, they are control animals. They didn't get infected, but the blue, the animals were infected. After day 4, you would see this animals started dying. And also the animal start losing body weight. They stop eating or drinking, so they start losing their body weight. On the left, you see it's all the uninfected control animals. Here, the animals are infected. They huddle together. They are lethargic. This is more likely day 3 or day 4. After that, they don't even want to go eat. So we have to euthanize these animals. So we have this model, and we are testing a number of drugs in this system. So essentially, it's almost, I'm getting to the last part of my talk. In the United States, after this vaccine, the scientists actually moved on. We are working with SARS-CoV-2, but also we are looking ahead for potential pandemic viruses. If you see, most likely the next pandemic would be caused by another RNA virus. It's hard to predict, but it's going to happen, mainly because the way we are now expanding and ruining the planet, there's a high chance we will hit another pandemic soon. So the coronavirus, we already know it's one, and also dengue, chikungunya, and also Nipah virus. Right now in Kerala, we do see cases of Nipah virus. These are ticking time bomb. We don't know when it's going to open up. So also, I just want to tell you one thing. We have Arun, and also I see Dr. Tripathi from Adolfo Lab. So we all know in the United States, before this pandemic, they didn't pay, Western countries never paid attention to infectious diseases. So it is one of the neglected field, main focus on cancer or cardiovascular diseases. However, there's a huge shift in the people's attitude now. There are major companies like Johnson & Johnson, Pfizer, Merck, they all in the race to develop a drug for these pathogens. So I think there's a huge shift now towards infectious diseases after the pandemic. However, for predicting the next pandemic, it's going to be, it's tricky. So currently we are working on multiple pandemic potential RNA viruses. As long as we are actually going deeper into the forest, we will get Nipah. Any virus actually in the jungle going to jump to the human. And also with the urbanization and pollution, we're going to have this kind of COVID or other respiratory pathogens. And with the polluted river and water, we're going to have mosquitoes and vectors. So we unless we care our planet, it's going to be very difficult. So the next pandemic could be a pathogen like a respiratory pathogen like a SARS-CoV-2, or it can be through food or vector. So one thing, this is my last slide, and one thing when we look through, there may be other pathogen or other pandemic. The rich countries are going to quickly bring them under control because of massive investment. The country like India, with 1.3 billion people, if there is another pandemic like a Nipah virus similar to SARS-CoV-2, a Nipah variant come out, what we do? So we really need to set up these kind of high containment facilities. At least each university should have a BSL-3 facility. And also we need to invest in BSL-4 facility, at least few of them throughout the country. So in case of a massive outbreak, we will be prepared. And also this is the time we should start preparing for next pandemic. So with that, we published a lot this year. It's a very productive time, and Arun is also part of our effort, and we have more papers coming. We also hiring postdocs. If any of the interested candidates in the audience, please reach out to me. This is the Los Angeles. It's a pretty city. And here is the hero of all the study, Gus Garcia. He has almost single-handedly done many of the projects I presented here. With that, I stop and take questions, or we can move to the next presenter. Thank you.
Thank you very much, Dr. Vaiti, for the wonderful and informative talk describing viral infection in different types of cell, coronavirus pathogenesis, and pandemic potential viruses. And we have a couple of questions, and concerning the time, I can ask you two questions. The one, Can you throw a light on the usefulness of lung organoids in modeling disease progression and disease candidate testing?
So the lung organoids, it's a very powerful system. However, it is the lab has to be very specialized. It's not like any lab can jump in and do it. You cannot get those. You need a distal lung tissue. So typically we get it from lungs which cannot be transplanted. So we have a big transplantation center. So those human lungs are discarded. So you could actually collect those lungs and collect those distal cells. You cannot get it through outside vendors. So however, it's very powerful to understand the disease mechanism.
Okay, so the last question. How is an RNA virus application affected by inhibition of a DNA repair pathway, for example, ATR kinase?
So it's a tricky thing, but you know what happens, the virus actually, when the cell getting infected, the virus wants the cells to think its DNA got damaged. So the DNA repair pathway kicks in. It started making more metabolites for fixing the DNA repair. So the virus somehow utilizes that mechanism for RNA replication. So it's still we are scratching our head, you know, why the viruses are doing that.
Thank you very much, Dr. Vaiti. And now we are moving to the second speaker. Thank you so much.
Thank you.
It's my pleasure to introduce the second speaker of the session, a clinician turned computational biologist who is best known for sequencing the first human Indian genome. His lab is involved in creating novel methods and resources for analysis and annotation of genomes and understanding the functional impact of genomic variants. He has been part of collaborative genomics projects aimed at understanding the Asian genome diversity. He is also instrumental in creating a comprehensive genomic surveillance program for SARS-CoV-2. He is a fellow of Royal Society of Biology. He has completed MBBS from Calicut Medical College and received his PhD from University of Pune. He is Dr. Vinod Skaria, a principal scientist from Institute of Genomics and Integrative Biology, New Delhi. Dr. Vinod will be talking about what did we learn from the SARS-CoV-2 genomes from India. I welcome you, Dr. Vinod, please.
Thank you, Dr. Arunachalam, and hope my slides are visible. Over the next 15 odd minutes, I will try to give a brief overview of what did we learn from SARS-CoV-2 genomes from India. And before that, I'll introduce the concept of doing genomics and genomics for surveillance. And of course, allude to a bit of the resources that you can access to learn more about these. Now, this pandemic has been quite remarkable in its ways. And one of the ways it has been remarkable that 400 years after discovery of the microscope, we have a new way to identify microorganisms which cause infections in humans. And what I'm trying to say is that contrary to the typical microbiological aspects of culturing, isolating, and observing an organism under the microscope, today we have a much better approach to look at organisms, and that is by sequencing the organism. And that is been possible because of developments in the last decade and a half, which has enabled fast, affordable, and scalable sequencing of humans as well as pathogens. And if you really go back to the paper which described the Wuhan Q1 genome, which is the first genome out of the pandemic, you would understand that no traditional microbiology was used in the isolation or identification of the pathogen, but it was entirely reliant on sequencing of the patient samples, which was the bronchoalveolar lavage. Now, this approach has a lot of advantages that it affords direct molecular diagnosis and at the same time offers characterization of the organism in terms of its context and its relationship with other organisms which has been sequenced before. Now, the pertinent question is why should we sequence genomes and why should we sequence these genomes over and over again? And that comes to the real point that the virus mutates, and in fact, all organisms mutate. The only difference is the rate at which they mutate. Now, for an organism like SARS-CoV-2, it's estimated that the virus mutates approximately 1.48 in 10 to the power minus 3 substitutions per base per year. That roughly translates to approximately 3 to 4 mutations per month. So theoretically, if you could space the sequencing of mutations at different time points, what you're essentially going to look at is additional mutations which occur in the background of existing mutations, or in other words, a chain of mutations, what we call as haplotype. And this haplotype can essentially give very interesting insights into how does the virus evolve, how does the virus spread, and also how does a particular variant or a combination of mutations emerge at a particular point in time. And this is sort of exemplified in this slide in here. Assume that you have 6 mutations, you could have multiple different combinations of mutations that occur, and the mutations occur in the background of an existing mutation or an existing variant. Now, assuming that all the viruses were equally able to mutate and equally able to diverge out and infect individuals, at any point of time, you should see equal proportions of each of these variants. But that's not what you really see in real world data. What you really see is some variants. In this particular case, a combination of mutations 1 to 4, we call it as a variant, could be found in, for example, a large number of individuals. Now, when you see such variants in a large number of individuals or a large proportion of the samples at a point in time, what you expect is that your variant has something abnormal. That's not really true always, though the popular perception is that in the cases QURP, it is all because of some variants. In fact, the commonest cause of such a variant to go up is essentially human factors, something like super spreader events, which could spread a particular variant far and wide. I'll, of course, give an example of how this has happened in India during early pandemic. Now, until around June, July last year, approaches to sequence large number of genomes to do such kind of analysis was not really possible. Of course, there were genomes that were done across the world, but not at the kind of a throughput which was possible to do it on a district-wide or a state-wide model, which included thousands of genomes which could be sequenced at one single go. And one of the earliest approaches to do this is what we call as COVID-seq. This was an assay developed by Illumina, one of the sequencing providers. And the advantage of the assay was that you could sequence anything from 750 sequences to around 3,000 sequences in around 24 hours. With appropriate automation and, of course, automation of data analysis, many of these tools which we were developed in-house, putting together open source tools, what we could put together is a scalable solution to look at genomes at a very large number, which would encompass states, which would encompass the entire country. And this assay, for the first time, was validated in a clinical site or a clinical situation in the lab of my colleague and collaborator, Dr. Sridhar Subbu, with, of course, a large number of young researchers contributing to this effort. Once we have validated this kind of an approach or an assay, what we went ahead was to collaborate with a number of states for which there was a depth of genomic data early in the pandemic. This includes a number of southern states, including Kerala, Karnataka, Maharashtra, Tamil Nadu, Orissa, so on and so forth. And the idea was to understand how this virus spread across these states early in the pandemic. And this was also an opportunity to collaborate with a large number of researchers and clinicians across the country who were interested in looking at the genetic epidemiology of SARS-CoV-2. And fortunately, this was also funded with a generous support from CSIR, though this was sort of an ambitious and a crazy idea. And what we really found out, looking at the genomic data from early pandemic, and this is also contributed by Dr. Divyadeep Saupati from CCMB, what we understood was we did identify a unique and distinct cluster of SARS-CoV-2 sequences, which did not have much of a representation across different lineages in the body. So this data from around May to June suggested that there are around 6 previously defined lineages or variants of SARS-CoV-2. And apart from that, one distinct lineage here marked in violet. And this seemed to have close similarity to what we call as the A3 lineage, which emerged out of Iran. So therefore, it was named as the A3I lineage, I to signify India, or it was also called the I lineage to signify this was a distinct lineage out of India. Now, of course, this genomic surveillance program could track the virus, the spread of the virus in terms of the proportions and the proportions over different points in time. And what we could understand is that this came out of a unique outbreak or a super spreader event, which has taken the virus far and wide across multiple different states. And of course, this was eventually taken over or bullied out by a much more infective virus, which is the A2A lineage of the virus, which has European origin. And this also paved way to looking at genomes at a much larger scale and to understand how did the pandemic spread across India in the early part of last year. And what we found was quite remarkable, that practically almost all major states had large outbreaks, and these large outbreaks were all traced back epidemiologically to super spreader events, either in marketplaces, in congregations, in marriage functions, and so on and so forth. And this also provided us an opportunity to work closely with a number of state governments to understand genetic epidemiology in close context, and also to build evidence to both create policies as well as to validate policies which are already in place. And one such example is Kerala. Now, Kerala is unique because it has a very large expatriate population, and following the pandemic lockdowns across different parts of the world, there was a large number of expatriates who were returning back to the state. And this was remarkable also in the context that Kerala has also one of the most draconian policies of quarantine of expatriates returning back from different parts of the world, 14 days to approximately 21 days at that point in time. And it was widely believed that the spread of SARS-CoV-2 in Kerala was through international travelers who were returning back to the state. Now, our genomic analysis largely suggested that almost all the cases that we could sequence from northern part of Kerala, this is Calicut and adjoining districts, came from 3 large clusters, and none of these 3 large clusters did trace their origin to a European or a Middle Eastern or an American variant. But largely, this clustered with variants which are just across the border. What it really suggests is that the largest spread of SARS-CoV-2 in the state of Kerala were from interstate travelers rather than from international travelers. So this sort of validates the policy that a longer quarantine period of around 14 days did significantly prevent the spread of the virus from international origin. But of course, the interstate travelers did not have to go through this sort of a quarantine procedure, and therefore they kept spreading the virus from across the state. And many of these could also be epidemiologically traced back in terms of the origin of the virus. Now, taking from here, Kerala did put together a program late last year, to involve such studies across all the 14 districts in a very systematic effort involving multiple tiers of the healthcare system. This includes the medical colleges. There were 12 government medical colleges, the state public health and regional public health laboratories, which are essentially testing centers, 2 university centers, the Inter-University Center for Biomedical Research of the NG University and the Central University of Kerala, and 14 district surveillance units, which are under the IDSP of the National Center for Disease Control. And this was partly funded by the National Health Mission in Kerala and CSIR through different grants. And what is put together was essentially a temporal map of how the virus spread across the state of Kerala, starting from early insights into the variant, which was called B.1.560, which had an extremely infective by nature, and also had a very peculiar genetic mutation called N440K in the spike mutation. Much later, of course, there was B1.1.7, now today called as alpha, which is spreading across the state, for which the state implemented active case finding and quarantine. But of course, much later, there was Delta, which started appearing sometime in March 2021, and the state implemented a policy of double masking, anticipating that this was a very infective variant. And much later, after Delta sort of gained foothold in the state went to an early lockdown in May with significant augmentation of testing capacity, beds, and oxygen supply. And as today, Delta has sort of taken over almost all the other genetic variants of SARS-CoV-2. Now, the real question that we're trying to ask is, are there newer genetic variants of Delta or sublineages of Delta? Today, there are at least 32 to 35 variants, and many of them have very unique mutations which are closely tracking. Now, such surveillance programs also allow us to understand very peculiar clinical context. In this particular case, we were looking at asymptomatic reinfections. Reinfections do occur, and now it's very well documented. But the quantum of asymptomatic reinfections were not really understood. And this was done with another center where we had implemented a routine surveillance procedure for healthcare workers, which included real-time PCR. And there were two individuals who tested positive on two different occasions. We call them as I1 and I2 for individual 1 and 2, and two episodes E1 and E2. And we could sequence the isolates, and we could suggest that they have very unique and distinct genetic mutations, suggesting that they were indeed reinfections of the virus. And the surveillance programs also have provided inputs into one of the earliest cases of breakthrough infections. This is actually the second published report of breakthrough infections across globally, where we had an opportunity to look at genomic the makeup of early breakthrough infections in the state of Kerala. And of course, such opportunities do exist, and they keep coming in terms of understanding the genomic underpinnings of very unique clinical presentations. And of course, today, now we also compile the data for the entire nation as part of the INSACOG program, as well as independent programs which are run by the states, and also independent programs which are also run by researchers across the country. And we have this unique dashboard which you can all access to understand how the virus is spreading across different parts of the country. And today, for researchers who are interested in integrating this data into their workflows, into their analytical pipelines, we also have the APIs available, and beacons conforming to the Global Alliance for Genomic Health, which is now deployed onto this service. And there are a number of now dashboards which have been coming up in the past few months, which has extensively used this API for federated learning and research. We also build and maintain a large number of key resources for SARS-CoV-2 analysis in India as well as across the world. This includes Phylovis, which is the updated phylogenetic visualization of all SARS-CoV-2 genomes from India. We also maintain two globally pioneering databases for immune escape mutations as well as functional mutations in SARS-CoV-2. And of course, as I suggested earlier, we do also mention and maintain the SARS-CoV-2 surveillance program datasets for the country. Moving on from here, the real question is, now of course we had Delta and we had a very bad wave, which has started subsiding across different parts of the country, including in Kerala. The real next question is, how is the virus going to mutate next, and what is it going to do? And largely, if you look at the genetic variants which have gained foothold across the world, you could see that the virus mutates, and fortunately, these are stepwise mutations, one at a time, or small steps at a time, which could either increase the transmissibility or the infectivity of the virus, or increase the immune escape property of the virus. So as we move forward, we are essentially trying to look at genetic mutations, which can sort of scale this landscape and try to understand how and where is the virus moving next. And this is also important in the context that if you could implement genomic sequencing in a very regular and consistent manner, you could identify emerging genetic variants very well ahead in time. And this is sort of exemplified also in this particular chart, which sort of maps Delta. So what you have in the top panel is essentially the cases out of the first and the second wave in Maharashtra. The middle panel essentially suggests the number of genomes sequenced at different points in time. And the bottom panel essentially suggests the cluster of delta and the time of origin of delta, which would be computed based on a concept called the most common ancestor. Now, what I suggest here is that delta from calculations suggests to have originated in July 2020. The earliest sequence of delta in public domain resources seems to be from around October 2020. That was around the time the first wave had happened in Maharashtra. And the first large-scale outbreaks of delta happened in Amaravati and Yavatmal sometime end of January and February 2021. Or in other words, if you had a very significant and consistent effort to sequence a large number of viruses as part of genomic surveillance, you had enough and considerable amount of time to be able to identify well in advance emerging genetic variants. It's not to suggest that you could prevent genetic variants from emerging, but you could understand the properties of an emerging genetic variants well ahead in time, provided you have access to data and access to genomes at that point in time. I think I'll stop there, but before that, also to suggest that therefore it's very important to identify such outbreaks across the country and sequence them very early. And therefore, you need techniques and tools to do this in an accelerated fashion. And one of the recent things that we have implemented is an accelerated protocol for sequencing SARS-CoV-2 on benchtop sequencers. These are essentially the MySeq and the iSeq sequencers, which are available across the country, very large number. And you could sequence anything from around 24 to 48 samples in less than 24 hours. And of course, this has been widely implemented in investigating many outbreaks in the country, across the country, as well as across different other regions in the Southeast Asian region, which includes Nepal and now the Maldives, where we have been working quite extensively with the WHO Southeast Asia office in Delhi. I think I'll stop there and take questions, but before that, let me acknowledge a lot of people who made this possible. Fantastic collaborators, Dr. Sridhar Chivasabu, who did most of the next generation sequencing, fantastic team members at IGAB and in my lab. And of course, this has never been possible without fantastic collaborators, early collaborations from the CCMP, Dr. Divya Chaupati, fantastic set of clinical collaborators from across the country, and of course, consortia, which includes the INSACOG, Indicogen, and the statewide surveillance programs. And of course, this is all possible only with considerable amount of funding. And thanks to all our funders, which includes early funding from CSR and continued funding from CSR for the statewide programs and the state national health missions, the WHO. Thank you very much. I'll take questions. I'll also have a slide for students if you're interested into looking at viruses in general. We have now an open program in Kerala, which was led by our lab, trying to track epidemics and use data science approaches to understand epidemics much better. Thank you very much.
Thank you so much, Dr. Vinod, for the very informative talk describing the genetic variability in SARS-CoV-2 and importance of having genomic sequences. We have several questions, but I can ask the very important two questions. So the one, why is there a TPR of almost 15 to 20% consistently reported from Kerala until yesterday, maintained in Kerala population? Is it because of local susceptibility of Kerala population?
Yeah, this is not to be looked at numbers in isolation, but actually culmination of multiple things that have happened together. One, of course, there is a significant large number of people in Kerala who were susceptible to infection because Kerala pretty much evaded the first wave which spread across the country. The second is that the state also has a considerable number of testing per capita compared to many other states. So it's very possible that a large number of cases, even asymptomatic cases, are now being detected. And third, of course, when you flatten the curve, the area under the curve is pretty much going to be constant, right? You could either have a peak that could exhaust the healthcare capacity and cause a large number of deaths, or you could flatten the peak. And flattening the peak necessarily means that you would have to move horizontally in time and therefore spread the outbreak over a large period of time. And that's essentially what Kerala has done it has spread the outbreak a very large period of time compared to the rest of the country. So at a point in time, if you look at, you cannot really compare the peak that has went through many other states in the country versus the number of cases which have been increasing, unfortunately, now decreasing over the last 3 weeks. So you cannot really look at numbers in isolation, but you will also have to keep the context and the fact also to measure here would be what is the death rate or the case fatality rate out of the pandemic, right? And Kerala, for that matter, has the lowest case fatality rate, almost nearing the infection fatality rate of SARS-CoV-2, which roughly suggests that almost all cases are being detected, and therefore the case fatality rate tends to become the infection fatality rate.
Okay. So we have one more question, that how one can get access to the INSACOG data, and have you discovered new variants in India?
Yeah, so the INSACOG dataset is available on the portal that is sort of suggested. Almost all, I would say almost 75% of the data is also available on GSAID, where you can download the data from. And the statewide programs have their own websites where you can access the data in that particular case. If there is any data that is not available yet, you could always request and get access to the data. But practically speaking, almost all data that is required in terms of genetic mutations, in terms of genetic lineages, in terms of the time points, in terms of the state of origin, they're all already available on the portal, and they're also available on the API. But if you're looking for sequences or FASTA files of SARS-CoV-2, yes, you could access it on GSAID, or you could access it from the state portals. Did you identify any new genetic variants in India? Many, right? We keep continuing to identify genetic variants, and genetic variants are, as I said, a part of natural evolution process of SARS-CoV-2. Nothing to be worried about all genetic variants. You need to be worried about genetic variants which consistently increase despite the best public health efforts. Yeah.
Okay, great. Thank you so much, Dr. Vinod, again.
Thank you very much.
Yeah. So now there's a third talk. I'm very much pleased to introduce the third speaker of this session, who is from Bangalore-based Indian premier institution, a virologist and immunologist whose lab is interested in studying host interaction of emerging human viruses, especially influenza, dengue, Zika, and coronavirus, to understand the molecular basis of viral pathogenesis. Also, his lab is exploring novel mechanism of antiviral innate immunity, which can be leveraged to develop broad spectrum antivirotics and improve vaccine efficacy. He did his PhD at International Center for Genetic Engineering and Biotechnology, New Delhi. Then he worked as research assistant professor and postdoctoral researcher at Icahn School of Medicine at Mount Sinai in New York. He is also a deputy of Wellcome Trust India Alliance Intermediate Fellow. He is Dr. Sheshank Tirupati, assistant professor in the Department of Microbiology and Cell Biology, Indian Institute of Science, Bangalore. Dr. Sheshank will talk about translating COVID-19 big data into clinical interventions. I welcome you, Dr. Sheshank, please.
Thanks a lot, Arun. Thank you to everyone at TIGS, Dr. Rakesh, Dr. Saibal, and thanks everyone in the audience for joining. So I'll talk about some of the work which we have done at our center here at the Center of Infectious Disease Research in IISc, where my lab is placed. And we actually started with establishing a COVID-19 diagnostic laboratory here back in March 2020, when the pandemic had just started to erupt. And by May, we were functional, and we started receiving samples for RT-PCR-based diagnostics. So we pondered that the samples which you are receiving, what else they can be used for? Can they be utilized for studying the disease and finding clinical interventions, prognostics, antivirus, things like that? So, I mean, SARS-CoV-2, everybody's aware, it needs no introduction. As Dr. Charles Rice, the Nobel laureate, said that we are in the midst of big virology education. So this pandemic has made pretty much everybody who has any connection with STEM fields some sort of a virologist. Everybody knows about SARS-CoV-2 and many other viruses now, which is very encouraging. And the virus, this one particular, has unusually long single-stranded RNA genome, which makes a bunch of proteins through different tricks. Some long polyproteins, which are processed into smaller proteins, some overlapping RNA segments, frame shifting. It utilizes multiple ways, which is not unusual. It is seen most often in viruses. They use tricks to maximize the coding capacity. One thing to note here is that we usually, or common people usually, hear about spike protein because that's the main focus of vaccine development or other structural proteins, but they constitute very small part of the viral genome. Majority of the viral genome codes for a bunch of other non-structural and accessory proteins, which primarily support viral genome replication, transcription, but many of them are dedicated to evade, confuse, and counteract the host immune response. And that underlies the pathogenesis of the virus, because the severe disease which you get upon SARS-CoV-2 infection, obviously virus infection and its inherent cytopathic effects are contributing, but in majority of the cases, the severe disease outcome is because of an exacerbated host immune response. So we'll get back to that. Now, what has also happened is, since the pandemic started, it attracted everybody from all aspects of STEM field, and they try to contribute in their own way to understand the disease and its progression. And a lot of data has been generated. And things are such right now that our ability to generate large-scale data has evolved a lot, but our ability to analyze all that data critically and gain insights is somewhat lagging. So this has led to the situation where we are really drowning in data related to COVID-19, but we are thirsting for the knowledge, which can be really useful. A lot of this big data is genomic surveillance, serosurveillance, a lot of virus host interaction studies on clinical samples or laboratory samples, and a bunch of other studies as well. One major aspect of these big data components come from these omics methods, proteomics, genomics, transcriptomics. And these mesh ball kind of Surrogate plots show that how many transcriptomics, RNAi, proteomics, CRISPR-screen, protein interaction studies were done back in 2020 December, and how that has increased rapidly by April 2021. It continues to grow. So all this data is out there, but which holds a lot of value, but someone has to take up the charge to analyze it properly and make sense of it. And that's what we decided to do. So we focused on omics data, which have come primarily from clinical sample and laboratory samples. And our focus was on two things. So I'll tell you what were those two things. So for COVID-19 management, these four pillars are most critical: diagnosis, prognosis, vaccine, and antivirus. Not just COVID-19. In case of any outbreak or pandemic, these four measures are required. For this pandemic, the diagnostics and vaccines have advanced astonishingly well. We have outdone our expectations. We have done very well. But the prognosis and antiviral development has been lagging. And we decided to focus on these two aspects and use our big data analysis approaches to address this. So first part we'll talk about is COVID-19 prognosis. Now, this figure here shows say infection and progression of disease and what are the methods which are currently in practice in clinic to assess the progression of the disease. So initially you'll get viral infection. Viral load should ideally taper down as the time progresses because the host immune response will build up. And as the disease progresses, you might have a very mild disease which may be taken care by your immune system, or it may progress to some severe form where you require hospitalization. In some cases, which varies between population and depends on the virus strains as well, ICU admission or ventilation will be required. In some cases, it might lead to disease of the patient. And these are the markers which are currently used: D-dimers, C-reactive proteins, production of IL-6, primarily in the serum. And these are measured to assess what is the progression of disease. But these are not exclusive to COVID-19. You can get these markers upregulated even in other infections as well. So other methods involve, say, imaging of your chest by X-ray or CT scan, which require you to go to the hospital and be exposed to more nosocomial infection, including more COVID-19 exposure. So the prognostic part is still lagging. And if we look more carefully how the disease progresses and in what cases it might lead to severity, so primarily it's a respiratory virus which enters your body through the upper respiratory tract, say your nasal cavity or through your mouth even into your trachea. And there it will infect the epithelial lining of cells initially, and they will immediately produce what are called interferons. Now these interferons are the cornerstone of our immunity, especially against viral pathogens. And then they will produce another battery of genes, interferon stimulated genes. Many of them will directly fight the virus at the site of infection. And many of them will, alarm the neighboring cell or the adaptive immunity and recruit cells which will sample the viral antigens and produce antibodies or T cell response. Now this follows a cascading event. So interferons are produced, then chemokines are produced, then interleukins are produced. And the yellow part shows here that if everything is in order, your infection will be controlled, you will have a balanced immune response. But as I mentioned, virus encodes for multiple proteins which interfere with this cascade of perfect immune response. And because of that interference, you might have an overridden, exacerbated immune response, a lot of inflammatory cytokine production, and that can lead to disease severity. So we argued that all of these events, this is starting in your nasal cavity where the primary infection has happened. And when you're collecting the nasal swabs from patients for COVID-19 diagnosis, in those nasal swabs, you should have some sample of those epithelial cells or other migratory cells in that cavity. And if you look into them in their transcript profile, you should be able to capture what is happening to the expression of these immune mediators. And there might be scenarios where there is a normal level of these expression, and there might be scenarios where these expressions might be upregulated and they may indicate that disease is progressing towards a severe phenotype. So what we are looking for? We are looking for immune mediators which are overexpressed in the nasal swab samples. And idea is that along with COVID-19 diagnosis, you should get prognosis as well from the same samples using RT-PCR method. So for this, what we did, we dig into the omics data sets which were published by the particular date when we started this analysis. And we chose these 4 transcriptomic data set and these 3 proteomics data sets. All of them are coming from nasopharyngeal swabs or bulbs. And they are all coming from COVID-19 positive patients or healthy controls. An idea of taking both transcript and proteomics data set is that we are looking for things which are supported by orthogonal data sets, both at transcript and protein level. So that will give us more confidence that something which is upregulated at both levels is truly upregulated and across multiple studies. So this was a meta-analysis pipeline. We had these 7 data sets. We sort of reanalyzed their raw data, applied our own statistical cutoffs, and followed by that, we did an cumulative score calculation, and which led to shortlisting of certain number of factors, which we then went on to validate in a COVID-19 cohort. So this is a number of genes coming from these different transcriptome and proteome studies and overlap among these proteome and transcriptome data sets. And the corresponding p-values are shown here as well. And a simple Venn diagram shows that this red circle contains all the transcript level upregulated genes. And the green and the blue circles show genes which are updated both at protein data sets or transcriptome and proteome comparison, at least in 2 studies. So we decided to focus only on things which were upregulated at least in 2 studies at both transcriptome and proteome levels. And that led to shortlisting of these factors, which are about 46 or 47. Then we looked at these genes, the shortlisted 46 genes, what kind of biological phenomena they are involved in. And our prediction was that these should be primarily the mediators of immune response. When you carefully at them, they are actually mediators of cellular migration, defense response to pathogens, interferon response as well. So it indicated that our analysis was in proper shape. And these are the list of those genes which were shortlisted by this analysis. And this diagram shows their upregulation level in different transcriptome and proteome data sets. And this bar here shows their cumulative upregulation. And what we did is that we shortlisted some top 12 or 15 factors out of these which had high cumulative upregulation. If you look among them, one set of factors which are S100s, they pop out very conspicuously. There were more S100s in this top 46 genes. So we took these top 12 and few other S100s which were out. We also took this gene called TIGS or thyrodoxine. I'll tell you why in the second part of the talk. And then we took these patient samples, the nasal swab samples, which we had received in our COVID-19 diagnostic facility. Half of them were COVID negative, half of them were COVID positive. And within those two categories, half were asymptomatic and half were symptomatic. Now, within COVID positive category, asymptomatic means no symptoms at all. They felt completely healthy. It's just that they turned out COVID-19 positive. And symptomatic means they had really severe symptoms, SARI-like symptoms or hospitalization or difficulty in breathing. And then in these nasal swab samples, we went on to validate the expression of these shortlisted genes. And as you can see, in positive symptomatic category patients, most of these genes were highly upregulated. The red color indicates more upregulation. We also looked at the corresponding viral CT value, and we found, at least in our data set, that the higher viral CT value corresponded with the more severe COVID-19 and as well as upregulation of these genes which were shortlisted. Now we looked at value of these genes in distinguishing different category of patient samples. So if a particular gene can distinguish between negative asymptomatic and positive symptomatic, it will be an infection marker, identifying the COVID-19 infection. If something can distinguish between negative symptomatic and positive symptomatic, it will be COVID-19 specific marker, meaning the above will indicate infection of any kind, whereas this one will indicate that the infection is by COVID-19 and not any other respiratory pathogen. And within positive symptomatic, positive category, the ones which can distinguish between positive asymptomatic versus symptomatic, meaning mild versus severe disease, these will be the severity markers or prognostic markers. And these statistical values show the performance of these shortlisted validated genes in distinguishing these different categories. And as you can see clearly that most of these genes, they perform very well in distinguishing positive asymptomatic from positive symptomatic category, meaning many of them had prognostic value. So we went on to perform what is called ROC analysis, which is used to sort of evaluate prognostic or diagnostic markers. And for these genes, you can look at, in ROC analysis, you basically look at the area under the curve of ROC curve, and anything above 75% or 85% is considered good, especially if you look at the S100 genes, S100A9, A6, A8, and S100P. All of them, they work very well together, and they are able to perform well in form of prognostic markers. And here we look at their individual performance at optimal CT cutoff and their sensitivity and specificity at those cutoffs. So you can see that S100A8 has sensitivity of 93% in identifying severe cases. A9 has 85%, others have 81 or 74. So what we identified in the end here is a gene signature, which is S100 family genes, and they can be measured by RT-PCR, and these are RT-PCR CT cutoffs. Anything below that will indicate severe disease, and with quite good sensitivity and specificity. An advantage of this signature practically would be that this will be measured in the same set of nasal swab samples which are collected for COVID-19 diagnosis, and you need the same infrastructure and manpower. By RT-PCR method, you can do this. So along with the diagnosis, you get prognosis also. You don't need to do any blood draw or visit a hospital for chest imaging. So this is how we try to translate the omics data into one clinical intervention, which is prognostic markers. What are S100 genes? These are primary mediators of inflammation. These are calcium binding proteins. Many of them are secreted, and they regulate cascading inflammatory signals, which was shown in the beginning, that they are mediators which connect initial interferon response to subsequent severe interleukin response, and precisely that phenotype these S100 family of genes they meet. Now we move to the next part, which is antivirus. So COVID-19 or SARS-CoV-2, as was mentioned initially, although the vaccines have progressed very rapidly, major kudos goes to the mRNA technologies and other plug-and-play technologies, but the antiviral research has lagged. But one good thing has happened, that now people have become more open to host-directed antivirals. So especially virus-directed antivirals, they require a lot of time for discovery and development. And there are a lot of host-directed antivirals which have value in terms of broad-spectrum antivirals, and people have opened up to them. And even virus-directed antivirals, especially the nucleotide and nucleoside analogs, they have broad-spectrum antiviral properties. They are also becoming more acceptable. But reality is, we don't have any specific antiviral for SARS-CoV-2 even today. Whatever is being used in clinical practices, effective or not, that's debatable, is something which has been developed for other viruses and being repurposed here, or is something which is actually targeting something in the host and not really an antiviral designed exclusively for SARS-CoV-2. Nevertheless, we have to accept these ways of interfering with viruses more, because pandemics can strike at any time, and antiviral discovery development is a very long process. So repurposing of available interventions is the way to go. So for this, what we did is, as I had mentioned, that there was another gene, thyrodoxin. This gene came up at the center of this Venn diagram, meaning that it was supported by all 7 datasets. In all 7 transcriptome and proteome datasets, it was found to be upregulated. So obviously, it was a high confidence factor, although it was not upregulated at very high level, meaning that as a diagnostic or prognostic marker, it did not have much value, but it was consistently found to be upregulated. Now, what is this gene? This is part of this thyrodoxin system, which basically adds and removes disulfide bond in target proteins, and it flips between oxidized and reduced state by the action of this enzyme called TRO, thyrodoxin reductase. And basically, this is a redox regulator inside the cell. Now, through this disulfide bond formation and breaking phenomena, it can regulate many processes inside the cell, including ROS production, inflammation, cell death, and proteasome functioning. And there are many well-known targets of this system, including the inflammatory signaling pathway, transcription factor, NF-kappa B, AMPK, and AP1 and ASK1. Now, a useful thing, we immediately found that thyrodoxin system can be disrupted by this drug called Auronophin, which targets thyrodoxin reductase enzyme, and this will lock the thyrodoxin in oxidized state. This is a drug which is available since 1980s. It is approved for use in treatment of rheumatoid arthritis. It is available in US by the name Redora. It's a gold drug, and it's manufactured in India by Cadila by the name Goldar. It's pretty cheap. It comes for 115 rupees for 10 tablet pack. And at least in literature, it has been shown to be effective against multiple infectious agents, even useful in treatment of cancer. But clinically, it is approved for treatment of rheumatoid arthritis. Now, since it was inhibiting the thyrodoxin system, we wondered whether or not it can have any effect on SARS-CoV-2 replication and pathogenesis. So we tested that. In cell culture system, first of all, we tested toxicity. It was soluble up to 2 micromolar dose. And then we used this drug in cell culture in HCK-ACE2 cells at reducing and looked at its effect on reducing virus replication. Here you see the data on subgenomic RNA production in infected cell in presence of drug. You can see there is close to 3 log reduction, and that is confirmed by plaque assays as well. So infectious virus production is also reduced. And as well as in Western blot, you can see that viral protein production, especially spike at 0.5 and 1 micromolar, is completely obliterated. And it has pretty good EC50 values, which are very well within the range which is used in say, human treatment for rheumatoid arthritis. Then we moved on further to validate its preclinical efficacy in a hamster challenge model. We tested the drug both in prophylactic and therapeutic setting. In prophylactic setting, we gave the drug orally to the animals 3 consecutive days followed by virus challenge. And in therapeutic setting, we started giving the drug post-infection for 3 days. On day 4, we sacrificed the animals and looked at different parameters. So here you are seeing the drug toxicity at 1 mg, 5 mg per kg. The drug is not toxic at all, which is not surprising considering it is approved for human uses. And here you can see the body weight of the hamsters during that 4-day period. The red line shows the loss of weight in control infected animals, and the blue line blue and green line show the treated group of animals, and they don't lose as much weight. Here we show you the viral RNA quantification in the lungs of infected animals. This is control group, and these are the treated groups. So both in prophylactic and therapeutic treated groups, so the viral replication is down at least by 1 log. Reduction in RNA copy numbers, little bit better performance in case of therapeutic regimen. And we did lung histopathology also, and there you can see that basically what you can see that this is a healthy lung phenotype. The drug treated groups, they mimic that. In case of infected control, there's a lot of infiltration of cells and edema. So the drug was able to reduce inflammation, reduce virus replication in vivo, as well as reduce virus replication in cell culture. We are studying the mechanism, how it's working. In the animals, at least, we looked at the inflammatory cytokine production, especially IL-6. We found that the production of IL-6 in the lungs was completely obliterated in case of the drug-treated group. So as mentioned, the drug is already known as an anti-inflammatory molecule, and its protective effect, at least in animals, it mediates targeting IL-6 production. And some would know that IL-6 is a therapeutic target. It's targeted by this monoclonal called tocilizumab, which is a very expensive antibody. But compared to that, this is a very cheap drug which works on similar mechanism. So this is the summary of the whole study. We started with multiple data sets, did a meta-analysis to shortlist things which are consistently upregulated. And then we validated those upregulated gene phenotype in a cohort of COVID-19 positive negative patients. And then we went on to identify a prognostic signature, especially the S100s, and also identified a drug which is effective both in cell culture and in the study. If you want more detail about it, it's published here, and IPs related to it are protected by SC. And all of these studies have been done by very talented. This is Avijit, who's an undergrad student. Wahida, she was a master's student. They led the study. And we got a lot of support from diagnostic lab here in CIDR ISC. And bioinformatic work, we got support from Dr. Dinesh Gupta's lab in ICTV. And financial support, multiple sources are contributing here. So ISC, Infosys Foundation, BIRAC, and welcome to CDR answer. Acknowledged. Thanks. I'll take questions.
Thank you very much, Shashank, for the wonderful talk highlighting the use of big data in identifying the biomarkers and identifying the therapeutic targets. We have several questions. We will take only 2. So, why is there a difference in interferon response in severe and critical conditions?
It's the doing of the virus. So virus interferes with our very early innate immune response. And most of the viruses, especially RNA viruses, which cause acute infection, it's very important for them to shut down the interferon response. And that's not surprising. SARS-CoV-2 does that as well. But something unusual about SARS-CoV-2 is that it promotes NF-kappa B pathway. So at one hand, it inhibits interferon induction. At the other hand, it sort of promotes NF-kappa B mediated gene expression, and that totally off balances the host immune response. That's the undoing of the host immune response which follows.
Okay. The second question is, what's the effect of non-pathogenic viruses against pathogenic viruses in prevention and therapeutic application without adverse effects and inexpensive?
Well, not really in therapeutics, it has not been tried, but obviously, if you look at the vaccine platforms, the Covishield vaccine you're getting is based on an on-pathogenic virus platform. So there it's finding more uses. We don't have viruses which attack viruses being used in therapy as of now, but there are bacteriophages, for example, people are trying them against bacterial pathogens.
Okay. Thank you, Shashank. Thank you very much.
Thanks a lot.
Now it's a time for panel discussion. Today we have a fantastic panel. Before we dive into the panel introduction and discussion, again, I request the participants to make sure that we will come away with a new idea, a fresh perspective, and a new strategy that we can apply to add more value to our research. Please post your question in the Q&A section for the panelists. Thank you all in investing your valuable time. Let me take a minute to introduce our first panelist. I feel elated to introduce our first panelist, who is a well-known molecular ecologist and population genetist from Bangalore, a woman whose lab focuses on revealing the process that drive patterns of mammalian genetic variation. Her research applies molecular methods in combination with computational techniques developed by her group for the analysis of modern and archival DNA. She's a part of a multi-institutional consortium effort on pan-India SARS-CoV-2 genome sequencing. She obtained her PhD from the University of California, San Diego, and completed her postdoctoral training at Stanford University. She's a Wellcome Trust India Alliance Senior Research Fellow, Indian National Science Academy Fellow, and also a recipient of Parker Gentry Award. That's Dr. Uma Ramakrishnan, a professor from National Center for Biological Sciences. I welcome you, Dr. Uma.
Thank you.
The question we have, this Bangalore Life Science Cluster, BLISS, is involved in developing efficient sampling methodology, disinfection technology, and so on. BLISS is one of the recognized centers in diagnosing and validating kits and other COVID-related research activities such as viral genome sequencing to identify the emergence of variants. Could you provide your insight on overall challenges BLISS experienced in the beginning and how BLISS overcome these challenges?
Thanks, Arun, for that question. I'll be very brief because I know we're running late. So BLISS, just for all of the listeners, is the Bangalore Life Sciences Cluster, which includes NCBS, INSTEM and associated institutions like TIGS, as well as CCAP. And, when the pandemic started and following the notice that government labs, central labs were given permission to conduct testing, me and several other colleagues together, we helped set up, initiate a pop-up lab to do COVID-19 testing. And it was a very different and interesting experience. A lot of commitment from volunteers across the campus, students, postdocs, and so on. And a lot of interaction with the Karnataka state, where we got all our samples from. I think the challenge for us as scientists was that we'd never done diagnostic style tests before. In science, our goals and milestones are long-term. We're used to research on the timescale of years. And to do something where we had to return results within a day was something which was a very good learning experience for all of us. And following that, we got involved in several research projects, as you may know, as well as the recent INSACOG-based genome sequencing. But through this whole pandemic, the spirit of BLISS has been that the campus should function safely, should contribute to society from a perspective of service, like testing or sequencing. We see these as service, and also science. So any new research we can contribute at this time is something which we thought is very important. Thank you.
We have two more questions. The second one, a rapid compressed sensing technology for COVID-19 pool testing was developed by your team. Could you talk on how you came up with this innovative idea and the advantages of using this test?
So I should correct you, Dr. Arun. So I was involved just more as a facilitator, but the computational approach for this was developed by a professor at IIT Mumbai, and he's now, he started his own startup company to look into this further, and also several of our colleagues from campus, Das and Sandeep Krishna. We all work together and try to look at whether this, basically when we do pool testing, we pool, and then if we have positives, we have to unpool and redo everything again. But in this compressed sensing, the idea is that you don't have to unpool, so you save time. And overall, the hope was that in this, using this kind of an algorithm, we can, very quickly figure out positive samples, even when the positivity rate is relatively high. So I think this method and the associated implementation is moving forward. And it was just an interesting idea to think of how computational approaches, in addition to molecular, we're used to thinking about, okay, what new methods can we develop, in the lab? But sometimes the analysis component can also be very, very important.
Great. The last question, being an ecologist, how do you relate losing biodiversity to the current COVID-19 pandemic?
So that's a loaded question. I guess it's a very difficult question. In some ways, as someone who's an ecologist, the onus is on us to really work out the details of what this means. You read about something like that in the newspaper and everybody will say it and agree with the statement. It seems like a truism, right? Like it is true, but fine, then what do we do about it? So how do we move forward in a way where we can minimize a spillover of pathogens, which will cause, a lot of human life, but also economic life and affect our societies. A lot, especially important to me, is that often the people who are most vulnerable are the most in society, don't have the ability to actually do too much about this, and they just have to face it. So I guess we must understand human-wildlife interfaces, how changes in land use will affect reservoirs and vectors, reservoirs like bats and rodents and birds and so on, and vectors, and how the dynamics of ecological change and people's interaction with ecology will result in spillover. Such understanding can help us minimize the risk, hopefully in the future. Thank you.
Thank you so much, Dr. Uma, for talking about the BLISS efforts and connecting losing biodiversity to the ongoing pandemic outbreak. Thank you very much. Now we are moving to the second panelist. I'm happy to introduce our second panelist, who is a co-founder and director of a lab in Bangalore. She has worked extensively on infectious diseases and maternal and infant health. She's also a trained antenatal and postnatal counselor and has worked in a charitable hospital. The focus of her work is developing scalable and accessible diagnostic technologies and wastewater surveillance of COVID-19. She received her PhD from University of Pittsburgh, where she also did her postdoctoral fellowship. That's Dr. Varsha Sridhar from Molecular Solution Care Health, which is ICMR report for COVID-19 RT-PCR testing. I welcome you, Dr. Varsha.
Thank you.
Varsha, we have a set of questions for you. That you have set up a citywide platform for longitudinal testing of sewage water and a dashboard to pick up location with the detectable coronavirus. Could you talk about your experience on this and the importance of the study?
Sure.
Actually, I think I should probably quote Dr. Vinod Skeria over here, who's talking about the need for surveillance, and Dr. Rakesh Mishra right in the beginning, who was also talking about wastewater surveillance here. I think surveillance for most epidemics or diseases so far, we've been looking at diagnostics as our primary way of surveillance, diagnostics of human cases. And I think COVID has kind of given us the opportunity to look at environmental surveillance. I was very fortunate to be part of a group called the COVID Action Collab, and we set up a citywide surveillance in Bangalore, where we test various sewage sources twice a week, and this has been going on for since over a year now. And I'm also very fortunate to now be working quite closely with Uma Ramakrishnan, Farah from TIGS, and Dr. Utpal Tatu from IISc, and we meet quite often to look at our data and to combine our data. Why I think this is important is that most of our existing surveillance, which relies on human testing, tends to be quite biased. You only test people who come to certain centers. You also tend to lose out on people who are perhaps migrating, or who don't trust the health system enough to actually come and get themselves tested. So when you have something like wastewater, it kind of goes past these human biases and just goes ahead and looks at, say, slums where human testing may not be very extensive, and or areas, like I said, where there are large migratory populations where you can't track back the people, and you might be able to get hotspots a few days before the actual community burden goes up. So you have a couple of days of advance warning with COVID, at least, is what we are seeing.
Great. So the related question, how does wastewater help with the surveillance of COVID-19 and other emerging infectious diseases?
With COVID, it's fairly straightforward. COVID does affect the GI tract, and so it does get expelled in the stool. There's also been some data showing that the stool samples do have COVID a few days before and a few days after it is found in the nasopharyngeal swab, though this data is a little iffy and it changes from study to study, I guess. In terms of other diseases, I would say that one of the biggest things that we are realizing that wastewater surveillance could be used for is in the terms of antimicrobial resistance. You do have antibiotic residues that are found in sewage water. And this is, I think, we think about, when you think about One Health, again, something which Dr. Mishra had talked about in his introduction, you think about One Health, and AMR is a classic sign of One Health, and wastewater surveillance is a classic kind of a tool to look at One Health. If you have antibiotics in your water, which you drink, you might be getting AMR genes through that. You also see AMR genes in the wastewater, and then that is also seen in the soil, that is also seen in the produce, and so on. So you can see a beautiful little loop in antibiotics and AMR. Other diseases that wastewater surveillance has been used for are other GI tract diseases. People have actually, originally, wastewater surveillance was used for polio in the 70s. And way back when, in the 1800s, it was actually used for typhoid diseases. So there are plenty of diseases actually that we may not even know about. In addition to infectious diseases, a lot of non-communicable diseases have also been tracked using wastewater in the research setting, not in the implementation setting. So for example, people have been able to find obesity hotspots. Looking at wastewater detection, they've also been able to find hotspots where drug abuse has been going on, things like that. So, yeah.
Wonderful. So the last question to you, this is related to the diagnostic. RT-PCR is considered as a most effective method for etiologic diagnosis of SARS-CoV-2 infection. So how emerging variants affecting the molecular diagnosis of COVID-19, and what are the common challenges in the pre and post-analytical stages for diagnosing coronavirus?
Sure. Thank you. And that's actually a fairly complicated question, I think. And many of the people here, many of the speakers here have been involved with COVID diagnostics. And I know that they would have very many thoughts about this as well. RT-PCR is considered to be one of the most sensitive things, but I think in community testing, the accuracy, the sensitivity is much lower than what we see in a laboratory setting. So at least in COVID, there have been many studies showing that the sensitivity is only about 60%. So you do have a fairly large population which are false negative, for example. We see this in the second wave as well, where, there were many newspaper reports talking about how the RT-PCR was negative, but the patient would come with a lot of signs of COVID. But the CT scan, not the CT value, but the CT scan would have a fairly high score for COVID positivity. So I think what the experience has taught me, at least coming from a basic science background and going into diagnostics, is that perhaps a single test is not a gold standard or like the be all and the end all. You can have a molecular test, but you do have a chance of it coming back as a false positive or a false negative. And then so you always have to correlate that with a clinical test, such as a CT scan, for example. There have been multiple innovations, of course, and we've all been reading about Feluda, other CRISPR-Cas techniques for COVID diagnostics. In India, I think the two innovations that really scaled and which have changed the way for point of care diagnostics has been the TrueNat machine and the CVNat machine. I was fortunate enough to help set up one of Bangalore's first TrueNat centers in a primary healthcare center. And it was very satisfying to be able to get a report in like an hour to a woman in labor or to a family with a dead body, things like that. So I think that those were all innovations that really, I think, changed the way diagnostics could be kind of made equitable, accessible, and at scale. A lot of paper-based diagnostics, I think, are in the process, and as are saliva-based diagnostics, and these would probably be emerging techniques. You had asked me about pre-analytical and post-analytical. And I think pre-analytical is fairly straightforward. Things like, temperature of starting off with the way by which the swab is taken, and to the temperature of during the transport. All these play massive roles. I'm not even going to go into things like RNA extraction, manual way, automated way, and now the CCMB protocol that came out a few months ago, which talked about how you could do RT-PCR without any RNA extraction, which I think has been also been a great game changer. Some of I think all the labs here, Dr. Shashank had talked about the IISc lab, Uma, I know, runs the NCBS lab, our lab. We all get government samples. And one of the major things that we struggled with a lot initially was that we would get these samples which were clearly not kept in cold storage at all. So you'd get lots of invalids. And many times you would have to give them out as perhaps negative because you would not be able to go back and track the patient and get a reswap. So those are the kinds of things that are definitely holes, I guess, in what would be theoretically possible and what is actually the difference at the ground reality. Post-analytical, again, interpretation of RT-PCR results is not an easy task. It does require trained people, and it does require the inclusion of various controls. This may or may not be, again, in large scale diagnostics, like in COVID, when you're doing something like 1 crore samples or whatever the peak testing was at one point of time, every day across the country, how do you know what are the controls that have actually been included, and how do you know whether the interpretation is happening concurrently? And finally, of course, the reporting. Very, very, I think, emotionally difficult thing for us was to be able to say, we have the reports, but then something happens in the reporting, either reporting to the patient or reporting to the ICMR. Initially, there were many cases where there were problems or difficulties in manual entry and things like that. And you'd invariably end up with a patient screaming at you because somebody down the line in the data entry had made a mistake, either at the ICMR level or at the reporting level. And then you'd have to have this huge feedback to go back and correct it. So thankfully, those post-analytical steps have I think, drastically been changed in the past few months with the introduction of automation. But these are definitely things that are valid, not just for COVID, but really for any diagnostics. I hope that answers your questions.
Wonderful. Thank you so much, Dr. Varsha, for explaining the importance of wastewater surveillance for COVID-19 and sharing your experience. Now we are moving to the third panelist. It's a great to introduce our third panelist, who is a virologist and molecular biologist who uses genomic tools to identify pathogens in brain infections. She's interested in studying novel and emerging human pathogens. She obtained an integrated MSc and PhD in life science from National Center for Biological Sciences, Bangalore, and was awarded a Royal Society Newton International Fellowship for postdoctoral work at the University of Liverpool, UK. That is Dr. Chitra Pattabhiraman, and DBT Wellcome Trust India Alliance Early Career Fellow at the Department of Neurovirology, National Institute of Mental Health and Neurosciences, Bangalore. I welcome you, Dr. Chitra. The questions we have, currently we have access to hundreds of thousands of SARS-CoV-2 genomic sequences and discovered many variants of concerns. Have you seen any specific variants that are particularly responsible for the brain infection?
Hi, thank you for that introduction, Dr. Arun, and also for the invitation. So with regards to, we know that for SARS-CoV-2 infections, you do get symptoms that resemble a brain infection. So you do get neurological complications. However, they seem to be from the studies that I have seen, and I haven't directly worked on this, they are not related to the virus itself. It seems to be an inflammatory process. Excuse me. So I think that while there are variants, they don't die. So unlike Japanese encephalitis virus, which is a virus that infects neurons, SARS-CoV-2 does not directly infect and kill neurons. That is not the main pathophysiology that we see, but rather it seems to be infecting the olfactory epithelium, microglial cells, and other parts, and causing an inflammatory response. So as of now, there is no strong correlation between variants and neurological disease.
Okay. We have the second question. You have carried out a study on introduction, spread, and establishment of SARS-CoV-2 in Karnataka by combining epidemiological and genomic data. Could you talk about the currently circulating Pangolin lineages in Karnataka based on SARS-CoV-2 genomic surveillance? Are there any concerning variants most prevalent in Karnataka?
Right, so thank you for that question. So the study that we did was actually in the initial part of the pandemic. And at that time, what we found was there were multiple introductions. There were multiple introductions of SARS-CoV-2 into the state. But from the recent data, so we are part, from NIMHANS, we are part of INSACOG. And the sequencing efforts of INSACOG basically have shown us that we have mainly Delta and sublineages of Delta circulating in Karnataka.
So the last question to you, the main aim of the Indian SARS-CoV-2 Genomics Consortium, the INSACOG, is to monitor the genomic variations in SARS-CoV-2 on a regular basis through a multi-laboratory network. Have you seen the pattern of evolving variants in Karnataka related to the data from all over the India?
Right, so I would say that roughly, as Vinod has already mentioned, we expect roughly about 2 to 3 mutations of SARS-CoV-2 every couple of weeks. So it is not surprising that the virus accumulates mutation on the existing background. So we do expect emerging variants, and if they go unchecked in the population, then we will see the emergence of new variants of concern as well. This is what we have learned, and it doesn't seem to be, I think that temporally, we have consistently seen differences in different states, but overall, the variants that take over seem to get everywhere. So if there is a variant of concern that emerges, we do expect that it will get around everywhere. So the challenge is actually to do this monitoring continuously and to focus on the logistics. So I don't know if that answers your question exactly, but the real answer is that the challenges are, the genomic sequencing is one part of the challenge, but the other part of the challenge is to get the data, that is the collection dates, all the metadata associated with it, in a way that this is interpretable, and then we can transmit it back to people who can take action. So the consortium sort of allows us to put that in context. So if we see a certain variation or mutation coming up in a certain part of the country. So for example, if I see a variant, let's say at 30% in some part of India, and I see it at 5% in Karnataka today, I will watch out for that and see if it increases over time. So because of the way we do this analysis and the way we are tracking, it is necessary for us to do it continuously and to keep track. So that is the challenge right now.
Okay, wonderful. Thank you very much, Dr. Chitra, for providing your valuable inputs on SARS-CoV-2 variants and genomic surveillance. Before moving to the concluding remarks, I request the chair, Dr. Rakesh Mishra, to provide his valuable comments and final statement. Dr. Rakesh Mishra, please.
Thank you, Arun. It was wonderful, and we got the latest updates in most of the crucial aspects of this pandemic. And as we see that although there has been tremendous progress in past year and a half, but it also shows that what are the challenges and how much more needs to be done to handle even a small virus like this when it comes. And there has been a tremendous collaboration and interaction, networking, which is something also obvious from, like you see, each speaker is talking about how different agencies, institutions, individuals have joined hands. And that's the way to do something fast and meaningful, not necessarily only meaningful, but also impactful at this scale. So I think we have learned from COVID as a scientific community much more than earlier, that how to respond to the requirements which may not necessarily be our own area of research. As Uma mentioned, most of us have never done diagnostics or anything of this kind. But if you see the contribution of academic institution in validating kits or developing new kits has been amazing. Most of the institutions have really diverted a lot of attention and got involved directly or indirectly in this process. So I think that brings a very positive thing, that when really we recognize the problem, we can make a difference and contribute to help the society. So, but pandemic is not yet over, although it looks like we are having the upper hand in most part of the country, and it is helping because already we have had a lot of infection in the past, so there is a reasonable number of seropositivity, which are the bigger cities, that is helping. And wherever it was not that much, like Kerala, we are still seeing something, but I think that's the way to go and try controlling as much as we can. And vaccine certainly is helping. We still have to continue, maybe for a long time, this culture of wearing the mask and avoiding, not giving chance to virus to play with us. A new variant will come only when we allow the virus to infect more people and spread. If we prevent that, then we actually can control this pretty well. And that's the thing which we have learned, whether it's airborne, whichever new things have emerged about this virus, but few things are very clear that how we can, how society has to contribute. It's not only the government or the scientists or drugs or vaccines. People have to get involved. Otherwise, even vaccines might not work if we keep letting the virus do experiments with us. So that is extremely important. And I'm really very proud the way Indian institutions have responded, and most demanding technologies. We say severe surveillance sounds simple, but to actually monitor it and go all the way up to variant analysis from the severe sample is, if you told 2 years back, very few people will take. Even we were surprised when we saw that actually it can be done. It's amazing that this RNA is hanging around for so long and can be used in such a meaningful way. So many things we have learned, all over the world, as our country, we have suffered hugely, got economic loss, and so many people, particularly poorer people, have suffered much more because of the economic consequences of this pandemic. But at least many positives have come, and we should take, if we learn from them and implement it. I want to repeat that we should not forget the origin of this pandemic from the, at least to begin with, from the bats, but a few other steps in between are not very clear. But we know that if we cross our limits, if we don't respect the wildlife, we are going to be in trouble. So it's extremely important to take this lesson for the whole society, whole civilization, that we have to respect nature and live with it and not own it and do it what we want to. And as a scientific institution, I think it has been really, in a way, a confidence booster that highly demanding technology, data handling, and even to large extent communicating, figuring out things, we have delivered whenever it was required and is continuing. So there are many positives to take. I'm very happy that speakers have touched the broad aspects in the domain they've been talking, and given the really the data from the lab, which proves the point and showcases how science can reveal information which can be implied and used by healthcare system and policymakers and the governments with such pace. So with that, I'm really happy, and I hope that these kinds of webinars and interactions continue, because that is one good thing that we have kind of adapted ourselves to during this pandemic. And because the spreading correct information and building public opinion about such pandemic and such situations is extremely important, and it's our responsibility as a scientist to be involved and not stay close in our lab or in our offices. So I'm happy that TIGS organized this and facilitated at least a small contribution to this large task that we have in front. When the new technologies are coming and new response is coming, people need to be conveyed. We should not take that whatever is given, people should take. They should understand. A lot of hesitancy to vaccine comes because we have not conveyed to people well. Otherwise, everybody is interested in having good health. Similarly, new technologies that are coming for genomics, gene editing, we need to approach a lot and be with people, because then only we will be getting first technology across and be an important support for science, because that's going to be important. When people are with us, then the system, politicians who work for people, they will also be interested in support. So I think it's a lot of implications, such activities, and I hope many such webinars will become a regular feature of our activities, and all of us will give part of our time to convey accurate information and message to people. So thank you all for spending your time and participating in this. And I'll hand it over back to Arun to say the final concluding words.
Sure. Thank you very much, Dr. Rakesh, for your fantastic statements. Okay, friends, now we are moving to the end of the session, extending sincere thanks to all the eminent presence here. First of all, I take this opportunity to express my deep sense of gratitude to the Director of TIGS, Dr. Rakesh Mishra, for providing his consent to organize this webinar, chair this event, and for his continuous support. Thank you very much, Doctor. I deem it a great privilege and honor to pronounce gratitude on behalf of the Tata Institute for Genetics and Society, and on my own behalf to all the esteemed speakers and panelists for accepting our invitation to present their important work and sharing with us their findings and opinions today to make this event a grand success. We are extremely thankful to Dr. Vaiti for his efforts towards understanding the host-viral interaction using cell organoid and animal model system. I'm very grateful to Dr. Vinod for providing his view on importance of sequencing SARS-CoV-2 genomes from all over India. I must mention our deep sense of appreciation to Dr. Sheshan for providing detailed information on translating COVID-19 big data into clinical interventions. Further, we are grateful to the panelist, Dr. Uma, who provided valuable insight into the genomic surveillance of SARS-CoV-2 and Bliss' role in disease management. Dr. Varsha for her thoughtful coverage for wastewater surveillance for COVID-19 and other diseases, and Dr. Chitra for explaining SARS-CoV-2 in brain infection. I would like to take this opportunity to thank motivated and dedicated colleagues and friends from TIGS for their suggestion and support. I'm very grateful to Group Leader Dr. Shaibal, Senior Scientist Dr. Fora, and Communication Coordinator and Vinod for their massive cooperation for organizing this webinar. I express my thanks to all the participants for your active participation and inputs. Again, I thank all the speakers, panelists, my colleagues, and participants for making this event great success. Thank you very much, everyone.